it changes its conformation by opening up the interface between two subunits, creating a crack or an opening in the ring-shaped structure. Importantly, this is big enough to fit double-stranded DNA through, and when double-stranded DNA binds to the sliding DNA clamp loader, it does so such that it is now encircled by the sliding DNA clamp. Importantly, only a DNA that has a primer template junction can actually fit within this region. Completely double-stranded DNA can't bend enough to fit in the sliding DNA clamp loading site. Also important is the presence of a 3' hydroxyl at the site of the ATP binding, which stimulates the ability of ATP to be hydrolyzed. So when ATP is hydrolyzed, this causes the sliding clamp to change conformations, release the sliding clamp and the primer template junction DNA, and this causes the sliding clamp to now close again around the double-stranded DNA portion of the DNA, and now it's ready to recognize the DNA polymerase and facilitate its processive DNA replication. Now at this point, I've told you about a lot of different enzymes, and I just want to tell you a little bit about how they work together as a replication fork. So the DNA polymerase III whole enzyme, this large multi-enzyme complex, actually does more than just synthesize DNA and load the clamps. It also stimulates the DNA helicase. And this is mediated again by that same tau subunit that is interacting with the DNA polymerase subunits. And this plays an important role because if the DNA polymerase I, either on the leading or lagging strand, becomes stalled, it will pull the Pol III whole enzyme away from the helicase, causing the helicase to slow down during the time it takes for the polymerase to restart synthesis. So for example, if it hits a lesion in the DNA that has to be repaired, the helicase won't run away at the same rate as it would if it were bound to the polymerase, because the polymerase is now detached. Once the polymerase can bypass that lesion or the lesion is repaired, it can catch back up to the helicase and the process can become very rapid again. I've already told you that primase activity is stimulated by binding to the DNA helicase, and in fact, if you modulate the level of interaction or the affinity of the primase for the helicase, you can actually change the rate at which it primes new syntheses. And so if it's faster, you'll make shorter Okazaki fragments, and if it's slower or lower affinity, you'll make longer Okazaki fragments. So the rate of Okazaki fragment formation is actually determined by this affinity. Finally, DNA polymerase III, as I told you, has its processivity dramatically stimulated by the sliding clamp, and in turn also by the sliding clamp loader, since that is required for loading it. So now that I've told you about the enzymes and how they work together at the fork, I'd like to take you through the events that are occurring at a replication fork one by one. So this is an illustration of a replication fork bound to the DNA polymerase III whole enzyme. You can see the helicase here with its unreplicated and unseparated DNA. The top is the leading strand polymerase. At the bottom is a lagging strand DNA polymerase. There's also a third polymerase, as I've explained, which is at this point in the reaction unengaged, and you'll see how it becomes engaged as we move forward. And you'll also note that there's large regions of single-stranded DNA here that are bound to the single-stranded DNA binding protein. And in fact, I've labeled this the trombone model of bacterial DNA replication because this loop down here actually gets bigger and smaller depending on where you are in the replication process, much as the trombone slide goes in and out as you play different notes. Now, I've shown you the SSD that's bound to the single-stranded DNA here, but for the rest of the illustrations, just to reduce the clutter, I've removed that. Now you'll note that there's a large single-stranded region adjacent to the helicase, and this is actually the perfect substrate for the primase, which will come in and synthesize a short primer in the single-stranded DNA region. And this is again mediated by the affinity of the DNA primase for the helicase. Now as soon as this is synthesized, it's recognized by the sliding DNA clamp loader as a primer template junction, and it then loads a sliding DNA clamp onto the primer template junction, making it now ready to be recognized by the unengaged DNA polymerase. So what happens next is that the polymerase binds the sliding clamp, associates with the primer template junction, and begins to initiate a second Okazaki fragment. Now I want to point out, during this process, the processes that I've been explaining, the leading strand polymerase continues synthesizing, as has the other lagging strand DNA polymerase. And in fact, once that lagging strand polymerase reaches the end of its single-stranded template, that is, the beginning of the previous Okazaki fragment, it will fall off the DNA, just as I explained, because it no longer has template, and it will become an unengaged DNA polymerase, just as the one that is currently making the second Okazaki fragment was unengaged at the beginning of the reaction. Okay, so we've taken you through this in slow motion. Let's look at what it looks like in real time. Okay, so this, you should now be able to label all these different subunits in this process. So you can see in blue here is the DNA helicase. Okay, and it's unwinding the DNA, both strands, and feeding it to two different DNA polymerases here. You can see the leading strand polymerase, which is immediately using the template on the leading strand, using the leading strand template to make the double-stranded DNA. And you can see its associated sliding DNA clamp here in the green. Now, you'll also notice that you see the arrival of a primase. Okay, here it comes right there, and it lays down a primer, which is immediately recognized by the sliding DNA clamp loader, which puts a new sliding DNA clamp on. And in this case, because the leading strand already has a sliding DNA clamp, this is immediately recognized by the lagging strand DNA polymerase, shown again in purple, and its associated sliding DNA clamp. Okay? Now, it may be a little hard to, actually, I should point out, there's one obvious difference between the model I showed you and this model, which is that there's only 2 DNA polymerases. And that's because at the time this animation was made, it wasn't actually known that there were 3 instead of 2 DNA polymerases. But that turns out to be very important, because as you'll notice, there's a period of time, each time a new primer is synthesized, and the lagging strand DNA polymerase has to recognize that new template, where there's no synthesis occurring on the lagging strand template. Now, in some organisms, this will be just fine, but in E. coli, the replication fork actually moves at 1,000 base pairs per second. And as you'll recall, that's the absolute maximum rate a DNA polymerase can go. So while the leading strand polymerase can handle that quite easily, the lagging strand polymerase couldn't do it if it had to come off and rebind all the time. On the other hand, if you have a third DNA polymerase, it becomes obvious how you can always have at least one polymerase, and often 2, acting on the lagging strand, allowing the overall fork movement to go at 1,000 base pairs per second, which is what's observed in vivo. Now, sometimes it's hard to understand how fast this really is, so let me give you a little way to think about it that's, I think, pretty impressive. So the double-stranded DNA is 20 angstroms wide, but let's just imagine that it's 1 meter wide. Okay? Now, in that situation, the nucleotides that would be incorporating would be floating around the room you're in at about the size of a textbook. Okay? And this replosome machine would be about the size of a FedEx truck. Okay? The sliding DNA clamps would be the size of very large eels. Okay? And what's most impressive is for a replication fork moving at 1,000 base pairs per second, that FedEx truck at the scale of the DNA meter wide would be moving at 375 miles per hour. So if you were standing in a classroom with a double hexamer tube of 1 meter wide going across the classroom, and let's say it's 50 feet across, that replosome would come through, spend about 0.1 second in the room, and change that 1 tube of 1 meter into 2 tubes before you would even really notice it. Okay? So these are really remarkable machines that are accomplishing quite a bit at a time. So I want to end by talking about a comparison between the bacterial replication fork, which I've told you about in great detail, and what we know about the eukaryotic replication fork. So there are corresponding proteins to all the proteins I talked about for the replication fork in bacteria found in eukaryotes. However, it's a little bit more complicated there. So instead of the single DNA polymerase 3 that's involved in replication at the bacterial replication fork, there are actually 3 different DNA polymerases that work at the eukaryotic fork. Pol delta is exclusively making the lagging strand DNA. DNA Pol epsilon is exclusively making the leading strand DNA. Now, the third polymerase is actually involved in the priming event. So while in E. coli, the primase is called DNAG and it's a single polypeptide, in eukaryotic cells, it's a complex between a primase, actually a 2-subunit primase, and a DNA polymerase called DNA polymerase alpha. And the primase synthesizes the short RNA primers and then immediately hands them off to the DNA polymerase alpha, which then extends them for a brief period of time before, in turn, allowing them to be taken over either by the Pol delta or the Pol epsilon leading or lagging strand polymerases. As I showed you before, there's both E. coli and eukaryotic sliding clamps. E. coli is called beta, and eukaryotic cells it's called proliferating cell nuclear antigen, or pCNA, and not surprisingly, this was originally identified just because it was prominently present in dividing or proliferating cells, thus the name tCNA. The sliding clamp loader is called the tau complex for reasons that you understand now in bacteria, and it's called the replication factor C, or RFC, in eukaryotic cells. Again, the helicase is a little bit more complicated story in eukaryotic cells versus bacterial cells. It's a single subunit repeated 6 times, the DNA G protein repeated 6 times in bacteria, but in eukaryotic cells, the helicase, the core of the helicase is the MCM237 complex, which is made up of 6 different subunits, MCM2, 3, 4, 5, 6, and 7, that form a hexamer with one of each subunit in the hexamer, so it's a heterohexameric protein. And it's not very active on its own. Instead, it's activated by binding to 2 other proteins, CDC45 and CHINS, that form the so-called CMG complex, which is the active replicative helicase. So I've told you already that there's 3 different DNA polymerases at the eukaryotic fork. We know less about the interactions of the fork as well, although we do know that Pol epsilon is held at the replication fork by interactions with the CHINS protein. In contrast, neither Pol delta nor the eukaryotic clamp loader is part of the replication fork, so that's very different from the prokaryotic fork. And this may be because eukaryotic forks move at a much more leisurely pace of 20 to 60 base pairs per second compared to the 1,000 base pairs per second. And so those lagging strand events that have to be tightly coordinated in bacteria can quite likely occur by just solution binding out of instead of being tethered immediately to the site of DNA replication, because there's much more time for that to occur. And in fact, for lagging strand synthesis, it probably occurs almost after the fork has gone by. So at this point, I hope you understand a lot more about how the replication fork works. And if you stay tuned to my next presentation, we'll talk about how you assemble these replisomes at sites of initiation and understanding how that's regulated during the cell cycle. So I just want to thank the people involved in this process. So Sarah Thornton did most of the animations that you saw. They were done in the MITx biology office as part of the development of a course that Sarah and Mary Ellen Wiltrout helped you develop called 728X, which is available on the MITx and edX platforms, and is a full molecular biology course talking about all the elements of the central dogma. In addition, the very high-resolution animation showing the E. coli replication fork in action was done in the DNA Learning Center, which is part of the Cold Spring Harbor Laboratory. Finally, I want to thank the Howard Hughes Medical Institute and the National Institutes of General Medical Sciences for supporting my research. My name's Steve Bell. I'm a professor of biology at MIT and an investigator of the Howard Hughes Medical Institute. And what I'd like to tell you about now is the mechanisms controlling discussion of the events that occur at the replication fork. Now, this process involves two key events. First, unwind part of the DNA to make single-stranded templates to assemble the ribosome on. And secondly, you need to assemble two copies of the ribosome because all genomic or chromosomal origins of replication initiate DNA replication bidirectionally, meaning that the ribosomes that are assembled move away in both directions from the starting site of replication. So the start sites of DNA replication are called origins of replication, and this is where the first single-stranded DNA is formed and where the first DNA synthesis occurs. They can fire with various so-called efficiencies, which is defined as the percentage of cell divisions in which an origin initiates DNA replication. Some origins have high efficiencies, others have lower, and I'll tell you more about that in a moment. Now, all the DNA that's replicated by the two ribosomes that are formed at each origin of replication, and a reminder, all chromosomal origins are bidirectional, that DNA is called a replicon. And this stems from a model proposed in 1953 by Jacob, Brenner, and Kazan for the mechanism of initiation of bacterial replication. It's a very simple model that composed only two components, the replicator and the initiator. The replicator is the DNA sequences that are required to initiate DNA replication. The initiator is a protein that was proposed to bind to the replicator and trigger the initiation of DNA replication. Now, the replicator, although it typically overlaps the origin of replication, is not the same as that. And it is instead all the sequences that are required. And this is sometimes a little difficult to understand, but think about it in terms of transcription, where the promoter is all the sequences required to correctly regulate initiation of transcription from the gene, but the transcriptional start site is a very specific sequence at which initiation actually occurs. In that analogy, the promoter is equivalent to the replicator, and the transcriptional start site is equivalent to the origin of replication. So, this is what origins are like, and replicators are like. How are they distributed across the chromosomes? So in E. coli, not surprisingly, where there's a single origin replication, the origins are 100% efficient. If an origin doesn't fire, that means one of the two daughter cells is not getting a whole chromosome. This is different in eukaryotic cells, where there are many, many origins. What's found is most origins do not initiate 100% of the time, but that's okay, because when they don't initiate, the replication forks on either side will replicate through the region that they would have replicated otherwise, because there's lots of excess replication capacity. It's important to note that any origin that doesn't initiate but is replicated is inactivated, so you never have an origin initiate after it's been replicated by an adjacent fork. Now, another interesting characteristic of eukaryotic replication is that when origins initiate is characteristic of different origins. Some will initiate early during the S phase of the cell cycle, the time in the eukaryotic cell cycle when DNA replication occurs. Others will replicate in the middle, and still others will replicate late in S phase. And it's sort of initially unclear why you'd want to distribute this. Why not replicate everything all at once? But it's more clear if you think about the fact that sometimes you'll have damage to your DNA. This could occur by a sudden change in the environment. And if you do, and you stop two forks, and if they haven't replicated the intervening DNA, the only way to rescue that situation is to initiate replication between those two forks and create two new ribosomes that can fill in that gap. And by reserving that subset of origins to initiate late in S phase, you will increase the likelihood that you can fix a gap that's caused by some sort of DNA damage in stalling of replication forks. Okay? So, we talked about origins. Let's talk about how initiation occurs. We're going to start with the initiation of DNA replication in bacteria. So this is an illustration of the E. coli origin replication. It has 5 copies of the so-called dimer repeats, which are the binding sites of the bacterial initiator protein, which is called DNA A, and 3 copies of the 13-mer repeat, which is an AT-rich, easily unwound DNA sequence. Now, when the DNA A is present, which is and active in the cell, and this is a highly regulated step in the process in bacterial cells, it will bind to the 9-mer sequences. This, in turn, forms a helical filament, which distorts the DNA, causing the DNA it's bound to have positive superhelicity, and the adjacent DNA to, in a compensatory fashion, generate negative superhelicity, which favors unwinding of the adjacent 13-mer regions. Interestingly, the DNA A that forms this filament can actually extend that filament by binding the single-stranded DNA rather than the double-stranded DNA, although it only does this on the top strand, as illustrated here, not on both strands. Now, at this point, we've accomplished one important event, which is to unwind the DNA at the origin, but we're clearly lacking much of the rest of the machinery, and the next thing we need to initiate replication is to load the replicative helicase. Single-stranded DNA so that it can gain access into the central channel of the hexameric ring structure. The third important property is that DNA C has affinity for DNA A, which recruits the DNA BC complex to the single-stranded DNA and allows it to now encircle the DNA, so it binds such that it will now encircle the single-stranded DNA. Now, importantly, not only does the DNA-DC complex load on the top strand that has DNA A associated with it, but it also is loaded on the bottom strand, but in the opposite orientation. So it will leave the replication of the origin in the opposite direction. So you can see that on the top it's going to move to the left, on the bottom it's going to move to the right. Now, we don't understand in detail how that second DNA-DC complex is recruited, although there are theories that suggest that DNA A, which also has affinity for DNA B, may use that interaction to recruit the second DC complex in the opposite orientation. Now, at this point, you'll remember that the DC complex is inactive, so the helicase is not active yet. Nevertheless, you'll recall that the interaction between the DNA primase and the helicase stimulates the primase to synthesize new primers. And that's the next step of this process. The DNA primase is recruited, binds to the helicase, and synthesizes a primer, and something about this process causes the DNA C subunits to release. Again, we don't understand that in detail, but that's clearly the step at which this occurs. And now you have active helicases, which can move to the right and the left, top to the left, bottom to the right, to unwind more DNA, so that you get an extended unwound region. At this point, we have two primer template junctions that can be recognized by DNA polymerase III holoenzyme, load sliding clamps, and then the sliding clamps can be recognized by one of the three DNA polymerase III enzymes. It will then initiate leading strand synthesis. Then, at some rate determined by the affinity between the helicase and the primase, a second primer will be synthesized. That will be recognized by the clamp loader and a second polymerase subunit. And then you have initiated both leading and lagging strand synthesis. And this is really the starting point that we discussed in terms of the function of the replosome during elongation. So you know what happens from here. Now, I want to make a couple other points at this point. You'll note that there was only a single sequence-specific DNA binding protein involved in this process. Most of the process is driven by either protein-protein interactions or non-specific or structure-specific protein-DNA interactions. So, for example, the recognition of the primer template junction is sequence non-specific, but very specific for the particular structure, that of a primer with a 3' OH available and an adjacent single-stranded region for the DNA polymerase to be engaged with. Okay? And that's true also for the primase